Hi guys. So in light of all the news stories this week about the Chinese scientist who's created the world's first gene-edited babies, I thought I'd do a little tutorial to try and explain this in a bit more detail. So firstly, what is gene editing? Well, as its name suggests, gene editing is a way of making specific changes to DNA, and this can be of a cell or an organism. And currently, genome editing is extensively used in agriculture, so our genetically modified crops, which are there to improve their yield and resistance to any disease or drought, for instance. And it's also used by scientific researchers in the lab because it allows you to make changes to specific genes, and then you can try and figure out how these genes are working and what effects the changes might make. So although in theory gene editing could be used to edit germline cells and therefore edit the genome of an entire organism, this is highly controversial, and it's currently actually against the law to do anything like this with human embryos. So at the minute, anything that you edit in a human embryo is only allowed to develop to 14 days, and anything beyond that is actually illegal. So this means that the recent news about the experiments that's happened in China has been a huge shock to all of the scientific community. So there are a few different methods that can be used to edit DNA, but I'm going to focus on what's happened here in China. So today I'm going to talk about the CRISPR-Cas9 method, and this is the one that they've claimed that they've used. So CRISPR-Cas9 was first discovered in 2012, and it's currently the most common, cheap and efficient system that's used for genome editing or gene editing in the lab. So CRISPR stands for clustered, regularly interspaced short palindromic repeats, and this is the DNA targeting part of the system. So it consists of a guide RNA, which is around 20 bases long, and it's designed to bind to specific DNA bases through complementary base pairing. So this means that you can target it essentially to whatever gene that you want to. So Cas9 is the nuclease part, and this acts as basically a pair of molecular scissors which can cut the DNA. So the guide RNA and the Cas9 enzyme bind together and they form this CRISPR-Cas9 complex, which can then go and seek out any target DNA. And then once it binds, the Cas9 enzyme will unzip the DNA and then cut the target DNA, causing a double-stranded break. And at this stage, the cell would recognize the damage to the DNA and it would try and repair it. So it would initiate lots of DNA repair mechanisms. And this allows scientists to use DNA repair machinery to introduce changes to the gene that has just been cut. So as I said before, this is a technique that is already frequently used in the lab. However, the reason that it's been such an outcry in the media this week is because this Chinese professor has basically claimed to create the world's first genetically edited babies. So these babies were twin girls and they were subjected to germline editing of their DNA, which is supposedly going to make them more resistant to HIV. So in this study, they took HIV positive fathers and HIV negative mothers, and in total they had 8 couples. So first what they did was they would wash the sperm, and this was to ensure that no HIV was present in the sperm. And then the sperm, along with a CRISPR-Cas9 enzyme, was injected into an unfertilized egg from the mother. So this CRISPR-Cas9 that they used was created to target and essentially remove the expression of the gene CCR5. And this is a chemokine receptor which is used by many strains of HIV to infect immune cells. So the idea behind this is that if you remove the CCR5 by CRISPR-Cas9, that you would then prevent HIV virus from using this receptor on immune cells to enter the cell and infect them. So in total, they were able to produce 30 fertilized eggs. 19 of those were viable. And then 2 out of 4 embryos, which were from one particular couple, were found to contain modifications to this CCR5 gene. So these were then implanted back to the mother, which gave rise to the birth of these 2 gene-edited twin girls. Now, one of these babies is said to have both of the copies of the gene missing, whereas the other one is said to only have 1 copy missing, which actually means that they still have 1 functional copy of the gene and therefore can still produce this CCR5 chemokine receptor. So it kind of doesn't make sense that this one would have been implanted in the first place, but they did it anyway. Okay, so the reason why germline editing like this is so controversial is because the changes that have been introduced will actually be permanent in all of the cells that go on to develop the whole organism, or in this case, that went on to develop the twin babies. So this means that the changes will also be present in their germ cells, meaning that they will also pass this change on to any future generations, which is a bit scary. And it's also actually completely unknown what the consequences of the loss of CCR5 will be. So this chemokine receptor may be important in other roles, and therefore the loss of it might actually be damaging to the twins. And today there are actually many other ways to prevent people from contracting HIV, or even treatments of people that have already got HIV. So such a drastic procedure seems kind of unnecessary. So if you'd like to read more about CRISPR-Cas9 and what's happened in China with the gene-edited babies, then I've included a few of the links that I used below this video for you to have a look at. And if you have any thoughts or comments on this, then please make sure you comment below. And as always, if you liked my video, then please subscribe to my channel to make sure that you don't miss out on any future tutorials. Thank you.